Hey everyone, we are looking at chapter 14 this week on replication, generalization, and the real world. So we'll look a little bit at some replication studies and what that means, the replication debate, a little bit about meta-analysis, and then replicability, importance in popular media, talking a little bit about external validity and generalizing our findings to other people, kind of looking at real world versus experimental settings. So again, the focus of this week is on repeating or replicating our research and looking at how that relates to generalization and the real world. So when we have something that is replicated, we are repeating it, right? To replicate is to repeat. So the results have then been repeated. It's not just hypothetical. We've actually gone out and repeated or replicated a study that we found previously. And we can do this in a couple of different ways. The most straightforward, maybe the easiest example to start with, is we can do what's called a direct replication. Basically, we are replicating exactly what was done before. We're doing the original study as closely as possible, trying to follow it to the T to see if we can repeat or replicate the results that occurred before. We can also do something called a conceptual replication. So it's not exact, it's not direct, but in this case, which is maybe a little bit more easy to do, the conceptual variables in the study are going to be the same, but the operationalization of those variables is going to be different. So we are maybe still measuring depression, right? We are just as interested in the effects of depression on maybe somebody's motivation. But the way that we define depression in our new study might be a little bit different than it was defined previously. Maybe we have people rate themselves on a scale of 1 to 10. Instead of in the previous study, they used an inventory like the Beck Depression Inventory. And maybe the way that we're studying motivation or operationalizing motivation might be different as well. So we're doing a very similar idea, right? The conceptual variables are the same, but we're going to define and operationalize them just a little bit differently. We can also do something called a replication plus extension. The nice part about this is we are going to replicate the original study, but then we add more. It's like, oh, we can find out that information, but let's also get a little more information while we're doing it. So we're going to add additional variables to test additional questions and see if we can add to what we already know. So if you remember, we talked about this example previously where people were studied on whether it mattered if they took longhand notes or notes on a laptop, and then how did they do on some questions? So we could add more information, like change the different types of questions from factual to conceptual. We can add another element here. But so when we do a replication plus extension, we're doing the same study, but then adding more so that we can make other conclusions and add to the knowledge that's already been found. Now, there is something called the replication debate in psychology, and of course, other sciences too. Sometimes we call this the replication crisis because we have some issues when it comes to studies being replicated. And if you click on this link here, it will take you to a website that's going to talk to you about how studies often fail on that replicability study, where when we do this largest replication study to date cast doubts on many published positive results. So don't trust everything you read in psychology literature. In fact, 2/3 of it should probably be distrusted. In the biggest project of its kind, I'm just kind of paraphrasing here, co-authors repeated work in 98 original papers from 3 different psychology journals to see if they independently came up with the same results. The studies they took on ranged from whether expressing insecurities perpetuates them to differences in how children and adults respond to fear stimuli to effective ways to teach arithmetic. So really studies on all sorts of different things. According to the replication's qualitative assessments, only 39 of the 100 replication attempts were actually successful. So what we saw was that despite the fact that they were repeating or replicating these studies, only 39 out of 100 of them were actually successful replication attempts. The 39% figure derives from the team's subjective assessments of successes and failures. So, what we see here is that the team found the average effect size of the replicated studies were about half. Only 36% of the replication studies found actually significant results, right? So, what we're seeing is that these studies that are being done are failing to get the same results a second time. So, we're having some issues with reliability and replication in these different studies that we're doing. Right, and I'll post that link for you so that if you wanted to read the rest of it. But this is a very common issue that we're seeing in the sciences, sometimes called the replication crisis, that even though we have found statistically significant results once, we don't always find them a second time. And sometimes the replication studies don't create or yield the same results as the original ones did, which is a problem. It's something has gone wrong somewhere. And some of the reasons why a replication study might fail is there might be some problems with the replication attempt. Maybe when somebody went to replicate that study, they had some issues come up, right? Maybe the effects were very contextually sensitive. So maybe it was something that was only able to be created once. It was like an isolated event. Something happened in the process of the original study or in the new study that wasn't able to be repeated. Maybe they didn't have a big enough sample size. The sample size was too small to yield statistically significant results. Or we can look at the number of replication attempts. Maybe if something was only replicated once versus many, many times, that might play a role in how likely you are to get a statistically significant result or a successful replication study. There might also be some problems with the original study. Again, sample size can be an issue. If you didn't gather enough participants, or this participants weren't gathered through, very statistically sound methods or research, methods that were sound, then you might not get the answers that you're looking for. You might not capture what's actually occurring. There's also something called harking that can happen, hypothesizing after results are known. Sometimes what we see is that people go back and they're hypothesizing and making hypotheses post hoc. They're making them after the fact, which isn't the best way to be making them. These hypotheses should be made previously, and then you're trying to gather that data rather than going back and making the data fit what you have come up with. Failing to report variables showing no effect. Oftentimes, we don't report a lack of statistical significance, and sometimes that can be just as interesting and just as useful as finding a statistically significant result. There's also something called p-hacking, where we run statistical analyses and we add or remove participants until a result is found. So sometimes what we see is, again, after the fact, the researchers are manipulating and kind of altering the data to find a result when maybe there wasn't one there in the first place. So when that result is found and published, the next person goes to repeat it or replicate it, and they might not find the same thing. There's also some problems with the publication practices. Journals prefer new findings and significant differences rather than null effects. So when we aren't publishing null effects, when we aren't publishing a study that said there was no relationship found, we're definitely getting a bias that can happen, right? Where we're only seeing the results that are statistically significant and not all of the articles or all of the studies where there wasn't an effect that was actually found. So we can make some improvements to our scientific practice to try and make this better. We could try overall to have larger sample sizes, right, to try and gather enough participants that our research will be sound and generalizable to others. And we could report all analyses and variables. Right? So this is something that there's been a lot of literature about, open science, right? Trying to create a better resource for sharing data and materials so that people could all take the same data and analyze it independently, and that information is kind of open and available to anyone and very shared. Also looking at the concept of pre-registration, where you have to pre-register the methods that you're going to use, what hypotheses are you going to have, and looking at some of those statistical analyses and data analyses before data collection, right? So trying to eliminate that p-hacking and some of that harking that can happen in order to make sure that the information being reported is actually consistent with the hypotheses and ideas that were proposed. We could also see that journals publish more replication articles or accept pre-registered articles, kind of broadening the scope of what most empirical APA style journals might be willing to publish. And then also looking at funding, right? Maybe better funding and better access to funding could also help to improve this practice a little bit as well. Another thing that we see a lot when it comes to replication studies is something called a meta-analysis. And this is a typical type of journal article where there's an incredible amount of statistical analysis occurring, yielding quantitative summaries of literature. So what you see is that people take a whole host of articles and studies that were done, and they take all of that data and all of the results, and then they analyze those results together to get one kind of giant meta statistical analysis. And they can make some interesting conclusions by looking at the data in this way. Now, it's definitely not free of limitations or problems. Sometimes with meta-analysis, you get what's called the file drawer problem, where a meta-analysis might overestimate the true effect size because those null effects weren't published. So again, we only have the data that's saying that these effects were present and not all of the different studies where maybe something wasn't found. So for example, in 2008, there was a study on antidepressants and depressive symptoms, and 38 out of 74 studies showed positive effects. 37 out of 38 positive effects were published, but only 3 out of the 36 negative or questionable ones were published. And so what you start to see again is this very much can skew the data. You're not getting the full picture of what's going on because we're only looking at studies where we have found an effect to be present. A few other thoughts here. External validity, another thing that we're talking a lot about here with replication, generalizing to other participants and other settings. So participants, population, not the population, right? So when we're studying something, we have to remember that, right? That it's a population, not the population. What do we need to actually generalize to the population as a whole, right? Did we take a probability sample? Was there random sampling that occurred, which we talked about previously? This is a very important technique in order to make sure that we can actually generalize the data that we've gathered. Settings where we can look at ecological validity, whether a laboratory study generalizes to real world settings is a question that we have to ask ourselves. If we conduct an experiment in a lab, can we generalize that to the real world, right? Is there a connection or relationship? Is it fair to say what happened in that lab is typical of what might happen in the world as well, or not, right? The questions we have to ask ourselves. Are we looking at a theory testing mode or more of a generalization mode with our study and with our external validity that we're analyzing? With a theory testing mode, when testing associations or causal claims, we're investigating whether there's support for a particular theory. Versus we might be looking more at like a generalization mode when researchers want to generalize their findings from a sample to a population of interest. So the lens at which we're looking at the data from can also play a big role in how much external validity we feel like we have with our experiment and with our study. One last thing here, with a little bit of the theory testing, I love this phrase, right? This is such a great acronym. We have a problem of using weird participants. And by weird, we don't mean strange or odd. Weird stands for Western, educated, industrialized, rich, and democratic, right? So most research in psychology has been conducted on North American college students. So what we see is that we're often using the same populations or same samples when we're conducting research. Western and educated, industrialized, rich, and democratic participants, which might be skewing or kind of altering our data a little bit. So also looking at the need to try and expand our participant base to get different demographics and try and get more representative data. So that is our chapter on replication, generalization, and a little bit about how that can relate to the real world. Make sure that you get going on all the other required weekly materials, and I will see you for our next topic next week.